Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit that supports human evolution research and shares discovery. Today, we tackle how a Neanderthal and Denisovan ancestry affects our lives and learn the important piece of the puzzle that underrepresented did genomes show us. If you're interested in the answers to these questions and hearing the latest in human evolution discoveries, start by subscribing to our channel and clicking the notification bell so you don't miss a thing. Let's welcome today's guest, Leakey Foundation grantee, Fernando Villanea. Fernando, thank
Hey.
you so much for being here with us.
Hi, thank you so much for inviting me. Thanks everyone for watching.
Fernando joins us from New York in the US, where he is collaborating on some exciting new research. He is based in Colorado, where he is assistant professor of anthropology at the University of Colorado Boulder. Fernando is a population geneticist who studies archaic ancestry in our DNA by looking at genomes from all over the world, which gives him a very unique insight into understanding the impact of this ancestry in our lives today. So, before we dig deeper, we want to thank the Ann and Gordon Getty Foundation, Camilla and George Smith, the Joan and Arnold Travis Education Fund, and viewers like you who made today's episode possible. If you're watching live, drop a comment or question for Fernando in the chat. If you're watching the replay, let us know what you find interesting and what you want to learn more about. So, advances in technology over the past decades has revolutionized studying genetics and allowed us to have access to personal genomics. People can see, for instance, the percentage of a Neanderthal DNA they have. But what does it actually mean to have Neanderthal genes present in our DNA?
Right. So as you say, this should not be a surprise to anyone that everyone who is around, who is alive today, has some amount of what is called archaic ancestry in their genomes. And so that is portions of the genomes that originate from either Neanderthals or Denisovans, or even some other hominids that we can identify, but we can't really link to any of the fossils. But we have those. And you would think that they are inert and they don't really do anything for your genome. But in reality, they actually really matter. So they impact the way we live our lives. They might impact our health. They impact what we call fitness, which is a biological quality. And so I'm really interested in studying exactly how these little genome fragments impact, everyone's quality of life, everyone's health, everyone's fitness.
Well, there are a lot of misconceptions about having archaic ancestry in our genes. How does your work disprove some of these?
Right. So we should start by talking about maybe the biggest surprise when it came down to studying Neanderthal ancestry in people's genomes. So Neanderthals, we thought were a mostly European species because most of the archaeology is centered there. That's where most of the fossils are coming from, and the tools, in the bones. Um, and then we went out and then sequenced genomes for everyone around the world and looked for Neanderthal ancestry. And we found out that actually everyone has some Neanderthal ancestry. And in fact, Europeans probably have less Neanderthal ancestry than some other populations. So people in East Asia, in South Asia, um, and even in the Americas have a high component of Neanderthal ancestry. They also have Denisovan ancestry, which is a completely different species. So these are pieces coming from two different sources that may be doing things that are different than what like the modern human variant is doing. So I'm really interested in looking at these.
One of the other misconceptions we hear a lot is that archaic ancestry makes you, less evolved. Again, like
R-
how does your work show that this is absolutely not the case?
Right. Yeah, no, that's absolutely a misconception. And maybe the best thing to think about it is you have to imagine that our species, so modern humans, right? We, like our ancestors evolved in Africa, in the African savanna, and that's a pretty lush environment. It's pretty wet. There is some subtropical areas with rainforest. Even if it's seasonal, like the image we're seeing here, like there's still a rainy period. So it's a pretty cozy place to evolve, and we are extremely well adapted to that environment. But while our ancestors were adapting to that environment, right, Neanderthals and Denisovans, they're evolving and they're adapting to the environment in Eurasia. And this is a much harsher environment, right? It's much colder, it's much drier. Subsistence strategies are much more difficult. And so their bodies are becoming really, really well adapted to surviving in these environments, right? So they're if anything equally evolved than modern humans at that point. They're just evolving for different challenges. They're doing different things. And when our ancestors decide to expand outside of the African continent and we run into these more harsher environments, like actually we are pretty misadapted for them. And it could be that the reason we are able to survive and thrive and expand is because we are admixing with these other groups that are really well adapted to these places. So there's nothing less evolved around these ancestry pieces. They're just doing things that are different because they are answers to different questions, right? Different problems in other parts of the world that weren't really the African continent.
Yeah, I guess what's some other examples of you know uh adapting to environment, especially like very extreme environments?
Right. So this is my favorite example, right? So I live in Boulder, Colorado, which is it's very high. So we're like 5,400 feet above sea level. Um, And once you're older, right, going upstairs gets difficult because there's not much oxygen. Like we actually have like only 84% of the oxygen that you have at sea level. So people that live in these high altitude environments have to adapt to, these difficulties, right? There's just less oxygen in general. And Boulder, and I will tell you, like it does get hard to go upstairs. So it's like pretty difficult. Like it's nothing compared to some of the more extreme environments on the planet. People in Tibet, for example, they are 12,000 feet above sea level, right? They have only like 65% the oxygen that you have um, at sea level. That is an extremely hostile environment for most people. Like your body can take some like short-term adaptations, like you can make more red blood cells to cope with this, right? So then you're carrying the same amount of oxygen in your body. The problem is then you're making your blood really thick, and that puts a lot of strain on your heart, right? And so it becomes really dangerous for you when you're doing some extraneous activities. In like the extraneous activity that any person can engage in is giving birth. It's like very extreme and it has a high risk of like going into cardiac arrest. So to adapt to living in these extreme environments, humans have evolved some strategies. But for people in Tibet, they have a variant of a gene called EPAS1 that really helps to make their blood stay very liquid, even though they need a high concentration of red blood cells. And the variant they have actually comes from Denisovans. Like we know it's a Denisovan variant that helped them to that extreme environment. We also know that Denisovans were probably somewhere around the Tibetan plateau. Like there is a jaw that is Denisovan that was found in that area. So through admixture with Denisovans, people were able to go into environments they wouldn't be able to go before, right, and solve some of these problems.
So fascinating. And I like the whole stairs thing. I visited a friend in Denver and I totally did not believe that that would be like a thing, but I'm like you do like you become winded pretty quick.
Yeah.
So one of the areas of your research is incorporating genomes from underrepresented populations. Why are these genomes so important?
Right. So before I was mentioning that before genomes, we thought that Neanderthals were a mostly European species. And so we thought, okay, this is where we're going to find a lot of the archaic introgression is going to be in Europeans. Um, that led to a lot of European genomes sequenced and a lot of effort going into looking at archaic introgression in Europeans. And we kind of sidelined all these other populations. But once we started to look at them, we realized that not only do they have more and more unique archaic ancestries, they're also probably solving some very different problems. And so that leads to some really, really interesting evolutionary stories. For me, I think like the most interesting evolutionary story is thinking of indigenous Americans, right, as they populate the American continent. Because when you think about it, like it's just an incredible journey and this incredible natural experiment, right? The ancestors live somewhere in, the near the Bering Strait, like the Bering Plateau. They are there for thousands of years. They are adapted to living in this kind of difficult environment. You can really farm, right? They probably have dogs, but they don't have any other domesticates because it's kind of difficult to keep them around. And then they make the crossing into the American continent. And all of a sudden, this opens all these possibilities, right? Because now they're running into all these different environments. They're running into the grasslands, they're running into deserts, they're going to the rainforest, they're going into the Caribbean islands. They're even going into high altitude deserts in Patagonia, for example, right? And so all of these involve new challenges. And so their biology has to adapt to these new challenges. And culturally, they also have to adapt to these new challenges. And not only did they survive, they thrive, right? They were like an incredible success story. Like these peoples and civilizations are extremely well adapted to living in all these different environments to the point where they invented like basically agriculture again. They domesticated hundreds of species. These complex civilizations, they invent mathematics, they invent astronomy, they build pyramids, right? So it's like it's an incredible story of success, but it's coming from a very unique sort of bottleneck in the past, right? They're all coming from this one population that is definitely not adapted to all these environments. And so I think that's fascinating. That is just my favorite, um, I guess, aspect of human evolution to look at.
Well, now that we've heard more about your research, let's hear about what inspired you to study science, especially as a population geneticist studying human evolution.
Right. So I had a pretty atypical journey, I think, to get to where I am right now. I was born and raised in Costa Rica. If you've never heard of Costa Rica, so we are a very, very small country, right? I think Costa Rica is like 0.3% of the continental landmass of the world, so it's very, very small. But we also have more than 2% of the world's biodiversity. So we have just this like concentrated biodiversity that is impossible to ignore. Like 50% of the area of Costa Rica is all still forest. And there's actually not that many Costa Ricans. It's only 5 million of us, so it's also a very small population. Um, and so I think for the time I was 6 or so, I decided that I was going to be a biologist. I was completely in love in biology. I was really lucky. My parents were really encouraging of this. And by the time I got to college, there was just like nothing else I wanted to do. And once I got to college, I was having the time of my life. Like these are the years that I remember like super fondly, because every class I took was so interesting. But also, I got familiar with field work, which is something that I never even considered before, right? So if you go do field work, it's really fun because it's like camping with a bunch of friends who are also like giant nerds. They're just interested in biology. So we used to go do field work, and everyone would spend their free time just like exciting things. The birders would get up super early and they would go look at birds. The botanists would just spend like their lunch breaks collecting plants and just like trying to, identify them. For me, I was really interested in frogs. And so I would go out late at night and I would spend all night just looking for frogs and then just sleep, uh, throughout the whole time I was still work. So that was, was really, really happy during those years. And then finally, that kind of took me into the direction of primatology. So I volunteered to work at a field site in Costa Rica, in La Pacifica, where they were studying howling monkeys. And that got me in contact with all these interesting scientists, some of them from the US. And through them, they encouraged me to first visit. So I visited ASU and got to meet Ian Stone. I visited Santa Cruz, where I got to work with Nate Dominy. And then I was encouraged to apply to grad school. So then I went and got my PhD. And it happened to be at the right time that genome, the technology to generate these human genomes was getting really, really good. And genomes are being generated like faster than we could analyze them. So it set up this environment for me where I didn't have to spend that much time in a lab because I did when I was a PhD student. I spent all this time in a lab trying to get these genomes, and it takes a lot of time. But all of a sudden, I didn't have to because these genomes already exist now, and I can just get them as files on a computer, right? I can just like anyone that has a web browser, you can go and download like the genomes data for free. You can download Neanderthal genomes for free. Like if you know what you're doing, you can just do all this on your own. And so that's what I do now. I don't go to the field anymore. I don't work in a lab anymore. I just work in a computer all day.
Well, we'll hear more about what it's like to be a population geneticist, but let's turn the virtual floor over to Fernando and hear more about his work. Before we do that, though, if you're enjoying this episode of Lunch Break Science, let us know by giving us a thumbs up or dropping a comment in the chat or making a donation on our website to help us create new episodes. Now, let's turn the virtual floor over to Fernando to hear more about his research. Oh, see you
All right.
on.
Thank you. So, okay, I picked one story that I want to talk about today. So this is one gene called MOC19, and we'll talk about how there is Denisovan and Neanderthal introgression for this gene and why that is important. Before I start, I do want to thank all my co-authors. So this has been a giant collaborative effort. So I want to thank everyone who is here in the slide for all their contributions to this project. Okay, so we've talked about this a little bit, right? So archaic introgression was really common, right? It's just a big part of human evolution. So if we think of human populations today, so this is maybe people in different continents today, and as we go down this axis, this is going back in time. So people in different continents today are just run into each other and to the origin for all modern humans. And then if we go all the way to the bottom of the graph, you see there's some other branches, right? In red, that's Neanderthals. In green, that's Denisovans. In yellow, maybe these other hominids that we also have some ancestry from, but we can't quite identify yet. And every one of these dotted horizontal lines, that is an instance of introgression. That is moments in which these populations are running into each other, admixing, their offspring are hybrids, right? And so that ancestry is being incorporated into populations going forward into the future. So that was extremely common. It happened all the time, right? And then if we look at what that means for a genome today, so this is a representation of maybe all of human genomes, right? There are 22 chromosomes and the X chromosome. In red, the little bars in red, that is Neanderthal ancestries. That is portions of the human genome in which at least someone alive today has some Neanderthal ancestry. And the same is true for Denisovan genes, so that would be in the little blue lines on that circle. So there's a couple things to notice on the circle. First of all, it's not complete, right? There are a little gray areas where there are neither these little red bars or the blue bars. So we call those deserts of introgression. Those are areas in which no who is today carries either Neanderthal or Denisovan ancestry. And what that means is that those portions of their genomes were doing something really different that probably wasn't great for our ancestors. So natural selection is eliminating those, right? So we know there is a force that is removing things that are not helpful. And then everything that's left either could be neutral, so it's not doing anything, but that's really unlikely. More than likely, it has a subtle effect that is actually helpful. And there's some just extremely helpful where it's really obvious that something important is happening. And so that's some of the examples that are listed inside the circle, including the top one, which is EPAS1, is the gene I was telling you about, that has helped people in Tibet adapt to an extreme environment. But we see a bunch of other examples for this. Right, and so the one I want to talk about today is MOG19, and I'll get into MOG19 in a minute. But I also want to talk about this gene in indigenous American populations, right? I was describing previously how I think this is like an incredible part of evolutionary history, right? So their ancestors who lived somewhere around Bering Strait, so kind of the middle top of this map, they didn't really run into Neanderthals or Denisovans in the American continent. As far as we know, there were no Neanderthals, there are no Denisovans in the American continent. But the people who moved into the American continent originally, they were already carrying that archaic ancestry, right? Because ancestors in Eurasia had already run into these archaic species. So their ancestors are running into this continent, right? They're moving into a new continent full of new and exciting environments and challenges. And they are spreading everywhere. They spread really fast, like it's been estimated, maybe in 2,000 years. They are all over the place, running these new environments, and they succeed. So they do really well. But I think that having archaic ancestry might have actually facilitated that biological adaptation. And the reason is because most humans, like most of the human genome, is actually really identical. Like most of the genome is really unvariable. Like most people who are alive today, we're really, really related. Um, but the archaic ancestry in our genomes is the part of the genome that is the most variable, right? Because these archaic variants are just really different. So that is fuel for natural selection, right? That is natural selection saying, hey, there's a problem. Maybe we can adapt by looking at these two different variants, and maybe one of them will do something interesting that will help. And so that is what I think happened in for a lot of these populations. In particular, this in MOC19 seems to be like that seems to be a pretty key part of that adaptation, right? So MOC19 is a mucin. Mucins help you make mucus. So MOC19 is expressed in the saliva, in your esophagus, it's expressed in your corneas, in your lacrimal glands. Basically, it forms gels. So every aspect of your biology that is kind of like, you know, slimy, that is MOC19, right? And these are mucins. They agglutinate these lipids and they make them kind of like thick, right? And so MOC19, when you look at MOC19 in humans, you see this really interesting pattern. So here, this is the genomes of everyone from the 1,000 Genomes panel. So this is a population, or this is a panel of people from all around the world in all these different continents. The little blue bars you're gonna see, that is just like different people's genomes for MOC19. And they are listed based on how similar or how dissimilar they are from a Denisovan. So the top row, going horizontally, right, from the left side to the right side, that is like how close or how far they are from a Denisovan. And if we look at Africa, for example, most of the people who live in Africa today, if you look at their MOC19, is very different from the Denisovan. So it's all the way to the right side of that graph. But if you look at the other populations, right, like South Asia, East Asia, Europe, or the Americas, you see that there is this little spike that is on the left side, right? That is very similar to the Denisovans. There are people who are carrying very MOC19 that is actually of Denisovan origin. If we look at the bottom part of that graph, it's the same thing on the bottom axis. So you see a lot of these dots are all the way to the right. That is people that are carrying the human MOC19, so it's different from a Denisovan. And then all the dots to the left, these are people carrying the Denisovan MOC19. And now on the vertical axis, that is distance from one of the Neanderthals. And the people who are really dissimilar from the Denisovan MOC19 are really close to the Neanderthal. The people that are really close to the Denisovan MOC19 are really different from the Neanderthal. And so that is confirmation that this variant of MOC19 is coming from Denisovans, not from Neanderthals. I should have done that earlier, sorry. This is just to highlight the individuals that are carrying the Denisovan MOC19 in these populations, so these red circles. Okay, so then if we look at out of the populations around the world, who is carrying more, who is carrying less of the Denisovan MOC19, we have this graph. So it's colored such that if you're carrying less Denisovan MOC19, you'll be in some of these cooler colors like the purples. If you're carrying more of the Denisovan MOC19, then you're gonna be approaching green. And if you're carrying a lot, you're gonna be in the yellows. And as we can see in the populations around the world, right, like people of African origin, they don't carry the Denisovan MOC19. People in South Asia, East Asia, and Europe carry some small amount of Denisovan MOC19. But if we look at the American populations, these are admixed Latino populations, right? So Puerto Rico, Colombia, Peru, and Mexico. You can see that a lot more people carry the Denisovan MOC19. And if you look at Mexico in particular, it's a lot of people that carry the Denisovan MOC19. So another way to quantify that is to just look at specific positions that we know are unique to the Denisovan MOC19 that is not found in modern humans. So you can see that the Denisovan has a specific, specific variants there. The standard color in sort of that cream. If you are a heterozygous, like two of these Neanderthals, you'll be in brown. If you don't carry the Denisovan specific variant, the cells will be in black. So if we look on the bottom half at some of these American populations, once again, Mexico, Peru, Colombia, and Puerto Rico, you see that people carry a different amount of this Denisovan MOG19, but in Mexico in particular, it's really high. So 30%, that is almost like 1 in every 3 people are carrying the Denisovan MOG19. So that is extremely high for an archaic variant. That means that this Denisovan MOG19 is doing something really, really important in these populations, and thus is found at higher frequencies. Natural selection is actually increasing the frequency of the Denisovan MOG19. We see in Peru, it's maybe 1 in 5 people, and then Colombia and Puerto Rico is a little bit less. But actually, the reason for this is because these populations are admixed to different degrees with Europeans and with African individuals. This was, as a result of the European colony and the African slave trade. And so populations that have a higher indigenous component of their ancestry also seem to be carrying more of the Denisovan MOG19. And to confirm this, I also look at some ancient individuals. So the top line of the bottom half of the graph, which is pre-colony Americans. That's me looking at some ancient individuals. These are individuals who lived and died before the European colony. Some of them are hundreds of years old, some of them are thousands of years old. And I looked at whether they have this Denisovan. In fact, they do. A lot of them do, like even to a higher frequency than people in Mexico. And that indicates that the Denisovan MOG19 is part of the indigenous component of ancestry in people living today. And it was selected for in these populations before the European colony. So for these ancient individuals, this is just like a quick map where the individuals come from in the different colors. And then the individuals are listed on that little square table on the right. Every individual that is highlighted in yellow, that is someone for which they are carrying the Denisovan MOG19. So 14 out of the 23 individuals that I looked at, they're all carrying the Denisovan MOG19 in all these different populations around the world. So this is, or around the Americas. So that's pretty incredible. That means that the Denisovan MOG19 is doing something really interesting, is really important for all their ancestors. I also want to take a minute to say thank you to the Leakey Foundation. This is actually the funding that I used to get these ancient genomes was coming from one of their grants. And this allowed me to confirm that the Denisovan MOG19 is actually doing something super cool in these populations, all the way back to even thousands of years ago. Okay, so now we have to confirm that it's actually natural selection that is doing something interesting here, right? It's not just like random forces that are making this Denisovan MOG19 more common. And I'm not going to go into a ton of details of what this means, but there's a specific test for natural selection you can do called PBS. So under PBS, you look at all these positions across the MOG19 part of the genome, and then you see if these positions are, more represented or less represented relative to some other populations around the world. So on this graph, if you see that dotted line, each one of these dots is a position on the genome on the MOG19 gene. If the dot is below the line, that is people that have the representation of that position. If it's above the line, that means that it's statistically overrepresented, which means natural selection is actually probably picking that up in a way that is different from anywhere else in the genome. And these are colored based on the specific ancestry. So these are all in living people today, but in Mexico specifically. And something that's super interesting is when we were looking at a small portion of MOG19, that's the little blue dots. Those are all Denisovan specific sites. So what I've been calling the Denisovan MOG19 so far refers to that little piece right there that is absolutely coming from a Denisovan. But once we started looking outside of that piece, we realized all the flanking regions, so the regions around it, actually match more to Neanderthals than to Denisovans. And that is going to be really, that was shocking and surprising, but it's going to be important in a minute because I think that reveals a little bit of how humans ended up with the Denisovan MOG19, and it wasn't like a simple, it was like a very complex story. So to tease that apart, right, I also wanted to look at heterozygosity. So heterozygosity is a measure of how different different parts of your genome are, right? So everyone is a diploid. We are all diploids. We have 2 chromosomes for every part of our genome, which means that if we look at both those chromosomes, right, one's coming from your biological dad and one's coming from your biological mom, and because your dad and your mom are presumably different people, these 2 chromosomes are going to be different to some degree. And in some people, these are really different. In some people, these are very similar. In some portions of the genome, there's a lot of heterozygosity. Some portions are there's very low. So heterozygosity is just a comparison of how these two different variants, how related they are, right? Are they really closely related? Are they really different? For MOG19, the human MOG19 and the Denisovan MOG19 are really different. So in this graph, this is looking at heterozygosity for people looking at both their chromosomes. If both your chromosomes look really similar for MOG19, you're going to be at the bottom of that graph. So the very first individual you see from left to right, where it says DEN, that's the Denisovan. So both the Denisovan chromosomes for MOG19 are really similar. Then you have the Altai Neanderthal. They're also very, very similar. Then you have the Chagaskaya and the Vinia Neanderthals. And these two Neanderthals actually have a really high heterozygosity for MOG19. And that is shocking. Um, then we look at some human populations, right? So the first few you see in blue, these are all people with African ancestries. And they carry 2 copies of the human MOG19. So they are somewhat different, but not that much, right? It probably caps at around 60 or 70 differences between the two. You see people like that all throughout the world. So all these other populations also carry a lot of the human MOG19, which are only somewhat different. Then in orange, you start to see people who are heterozygous. They carry one human, one Denisovan MOG19. And these two different variants are so different, right? They're carrying something like 200 differences, right? So they're completely different. They're completely above everything else you see. You might also see some little green dots. Those are people who are homozygous for 2 Denisovan MOG19 haplotypes, right? So these are people you find mostly just in American populations, specifically in Mexico. MOG19 is so frequent in Mexico that you see a lot of pairings of 2 Denisovan haplotypes. And these are very, very similar to each other, right? And so that's why they're at the bottom of the graph there. So that was interesting in a number of ways. First of all, because it highlights that the Denisovan MOG19 is really different from the human MOG19. And the other interesting aspect of this is that 2 of the Neanderthals seem to be heterozygous, like they also have the Denisovan MOG19 somehow. And so we looked at those Neanderthals. So we did this really cool test, which I think we dubbed the pseudo ancestry painting test, in which we took the Chagaskrai and the Vinia Neanderthal. So those would be that teal line in the center of the graph. And then we said, okay, if they're truly heterozygous for one Denisovan MOG19 and one, the more human or maybe the Altai Neanderthal MOG19, that means that we should be able to reconstruct all these positions perfectly if we ask a computer, hey, pick one position from a Denisovan, pick one position from the Altai Neanderthal or from an African human, and see if you can reconstruct perfectly what the heterozygous position you see in Czechoslovakia and Vinia. And if that's the case, then paint it in a color, in this case, teal. If that's not the case, if you can't do it, leave it white. If you can only do it for one of the 2 halves, do it either in purple or in yellow. And what we find is that for over 96% of all these positions, these Neanderthals are perfect reconstruction, right? You can perfectly reconstruct their MOG19s if you take one position from the Denisovan and one position from the Altai Neanderthal or from one of these closely related humans that carry the human-like variant. And so that is fascinating because that means that these 2 Neanderthals also carry the Denisovan ancestry as well. And so we think this is maybe the most likely scenario for MOG19, right? So there's a Denisovan variant that is very different from both the Neanderthal variant and the human variants, which are more closely related. That Denisovan variant actually jumps into Neanderthals. It gets nested in the Neanderthal genome. And then some of these Neanderthals that are carrying the Denisovan haplotype actually admix with humans. And now we get the Denisovan model, we get it through a more complex route. So we get it through Neanderthals as opposed to getting it directly from Denisovans. And that's why you see it in all these other global populations. Then once the ancestors of indigenous Americans move into the American continent, they're carrying the Denisovan MOG19 and the human MOG19. maybe they're running into an environmental challenge for which the Denisovan Mag19 is uniquely suited for. And so all of a sudden, positive selection just kind of takes it and runs with it, right? And so then you see it at really high frequencies. Um, and so the last piece of the puzzle is to understand, what is it about this Denisovan Mag19 that is different from the human variant? How come, it's maybe being selected for? Maybe it's doing something special and different. So we, for that, kind of have to think of what mucins do and how Mag19 works. So all mucins work in more or less the same way. They're protein scaffolds. They then get bound with sugars, and the sugars themselves are what makes um, these mucins be more slimy, right? They just stick to things. So it's very sticky. In other mucins, you can have different variants of mucins that are different, not just in their coding regions, but also in these repeats where they bind to sugars. So these are sugar binding domains. And in MOC19, you get a repetition of a bunch of these domains. These domains are small, they're 30 base pairs. Um, and then just like they repeat it, and they repeat it in a lot of different copies. So for example, the human version of MOC19 has on average like 280 something copies of this. But in these American individuals today, like in Mexico, you see people that are carrying like 800 copies of this repeat. So the protein is much longer. It bounds to almost twice as many of these, or more than twice as many of these sugars, which will make the chemical properties of this mucin different, right? They will be sticking to things differentially. They'll either be more sticky or less sticky. They might just be sticking to different things. Um, and so once we quantify these copy number differences, right, we're looking at these populations once again, going from left to right. Left is people with African ancestries. We see they're carrying the human MOC19, and while they're variable in the number of copies, they seem to hover around like 280. Some people may have as many as 500. Once we go into these non-African populations, we see orange dots. These are individuals that are heterozygous. They're carrying one Denisovan Mag19, one human Mag19. We see that they tend to be towards the higher numbers of copies. The Denisovan Mag19 carries a lot more copies than the human one. But that is completely goes to the extreme once you look at like individuals from Latin America, right? Peru, Mexico, Colombia, and Puerto Rico, you see that people who carry the Denisovan Mag19 either in heterozygous form or in homozygous form, they have a lot of these repeats. Like the green dots are people who are carrying both copies of the Denisovan Mag19, and they're all the way at the top of this graph, right? They're carrying something like 800 copies, like more than twice as many as the average for people in other parts of the world. So we know that this longer protein is what's being selected for, like it's doing something interesting, different, but we don't actually know exactly why or what it's doing in a functional sense or a medical sense. Actually, the reason why I'm here now is because I am planning experiments on how to address this problem. Um, but to conclude, let's just to recap real quick. So there are two haplotypes of MOC19. They're really different from each other. So once you have them paired up in an individual who is heterozygous, they have really high heterozygosity. We see that the Denisovan one is coming from Denisovans into Neanderthals into humans. So that is interesting. That means that Neanderthals also got to keep it, and it's probably doing something cool in Neanderthals as well. But we also see that positive selection really favors this high copy number variant in humans and to the extreme in these American populations, right? We don't really know why, like what they're doing. We think that the two different variants might be sticking to different things. So some other mucins are part of the immune system, right? They stick to different bacteria preferentially. So it could be that the Denisovan mucin is sticking to a specific pathogen and the human mucin is sticking to a different pathogen. And maybe this became really important while you're just moving really quickly to the entire American continent and you're going to all these new environments. But we don't really know yet. So that is, um, a lot of future work, a lot of future directions. So that is all for this part of the presentation. Thank you so much.
Thank you so much. I have to ask, what is a day in the life of a population geneticist like? I feel like it's very different than anything we've heard about before.
Right. Okay. So, unlike all my other colleagues who either do field work, so they spend a lot of time in the field, or my colleagues who work in a lab and they do all these experiments, I think my day is probably very different from theirs. Like my day starts and ends on a whiteboard. So I will go to my whiteboard and I will say, okay, I have an interesting question, an interesting problem I want to solve. And I have access to all this data already exists on my computer. So how can I address this problem, right? And so the first thing I will do is I will draw on from theory. So population genetics is a field that is 200 years old. So we have 200 years of theory to draw from. And I will say, okay, so I think that this cool evolutionary process is happening, and this will result in, very specific things on a genome. So what are my expectations? And expectations are everything in population genetics, right? So I will say, okay, if this really cool process is happening, what I expect to see is this. If this process is not happening, I expect to see something else. So that is the start of my day, right? I start by just writing down what are my expectations. Then I will have to think of how to actually go about doing these tests. So I will move to a second part of my whiteboard and I will say, okay, so I need to look at these very specific aspects of evolution. Um, who has worked on this before, right? Because there's people that have created tests for all these different things. And so I will say, okay, I'll do something called pseudocoding, which means that before I type code that goes in a computer, I will just write it down in words that everyone can understand, right? So like just simple logical steps. Like I'll say, okay, I need my computer to first access all the genomes and then just cut out the part that has MOC19, for example, specifically. Then once you have those that you can compare them, I want to look at heterozygosity. So then I will say, okay, so how do you look at heterozygosity? Okay, so you go position by position and you ask the computer, I want you to compare every two people and tell me if they're different at that position. If they are different, just count it and just give me a count of how many people were different at that position. And then just give me an aggregate count of these things. So I will say that in like human words, and then after that, I will go to my computer and I will code that in like, I'll script it. I'll have my computer do it. It will take a long time because genomes are huge and this is a lot of information. Um, and I will get some kind of result, right? So that's going to be like some number. So heterozygosity, I'll get a number for that. Then I'll go back to my whiteboard. I have a big whiteboard, so there's like a third section of the whiteboard, and then I'll say, okay, so I see this number, and this number falls into expectation A, which means, okay, this actually proves my theory. That's great. Now, how do I go about making that sound interesting to other people? And so I will then think about how to present this evolutionary problem in a way that is approachable for everyone, how to explain the test that I did in a way that is approachable, in a way that is replicable. So anyone can take my notes and just do the same thing that I did, and they can confirm my results. Um, and then I'll think, okay, how do I make this approachable, not just to my colleagues who are going to peer review my papers, but also to the broader audience? Like, how do I make this interesting to people that care, right? Because ultimately, that's what I want to do, right? I want to tell stories to everyone so people care about them. So that is more or less what my day looks like. I mean, really, like this process takes years from beginning to end, but
Um, what are like this? Like, how big are the data you work with? The sets. I mean, they've got to be pretty huge.
oh yeah, no, they are really, really big. So, the human genome is 3 billion base pairs. Um, and even though, like every person who's alive is 95% identical on their genomes, the 5% that is variable, that's still 150 million base pairs of differences. Um, and so if you look at the 1,000 genomes data, which is what you're seeing on the screen right now, these are files from my computer. Chromosome 1, which is the largest chromosome in humans, um, compress into a type of file called a VCF file, which is a file that just like minimizes the amount of space. It's still 1.2 gigabytes. Once you uncompress it, it's like 60 gigabytes, just to look for chromosome 1. Now you're seeing what happens when you look inside one of these VCA files, right? It's just lines of basic text because that is the simplest, cheapest way you can do this, right? And so now you're scrolling down, looking at all different positions of the genome, starting in chromosome 1. And the human genome is so vast that you could scroll down for the rest of your life and you will never get to the end of this file, right? Like I can visualize this file all at once. If I had my computer to open a genome file like this and just like show it to me whole, my computer would probably burn down. So these are like enormous files.
That is just like it's so and like it's so interesting to see like how many lines. I'm like, do you think in numbers? Like
I mean, I don't think in numbers, right? I don't think in my ones and zeros. But this is why like your theoretical training as a population geneticist just comes into play, right? Because like when I go to my whiteboard, I have to find a way to ask a computer to do something that I can't supervise, right? Because I can't really see it doing it, like the files are too big. So I just have to say, okay, well, the logical steps are you open the file, you look at this portion, then you count this many things, and then you give me a number at the end. And then I have to trust that it's doing it correctly, right? Because the files are so huge that I can't look at them. Like my computer's not, like my brain is not a computer. Like if I open these files, like it won't really make any sense. So that I would say is probably the most challenging part of being a population geneticist.
oh, I bet. One thing I want to give a special shout out to an anonymous donor or a viewer who donated $50 to Lunch Break Science. We really appreciate your generous support, and just want to let you know that your donations go directly to making new episodes of the series possible. You know, and while we appreciate donations, we also like really appreciate you liking or subscribing to our channel, because that really again lets us know that you like what we're doing. So I have one more question before we get to our questions from our viewers. One are you've reached one of your dreams recently, which was to be able to extend PhD to students who traditionally wouldn't be able to get access to PhDs. So do you want to talk a little bit about that?
Yeah. So one of the difficult things about being a Costa Rican biologist, when you're really passionate and you have access to like this biodiversity that you want to study, is that we didn't really have a PhD granting program when I reached the end of my undergrad. Um, and so me and all of my friends who wanted to get PhDs, we just had to go different places, and we ended up like everywhere around the world. For me, I was really fortunate that I had individuals that would just guided me through the process of applying for a PhD in the US, which like if you're coming from outside the US can be really daunting. And because of that, I was able to just do what I love, right? And so one of the things I really wanted to do is I wanted to pay that forward. I wanted to be in a position where I can actually extend invitations to people to get a PhD. In my institution, in my department, Department of Anthropology at CU Boulder, they've been extremely generous, right? They encourage capacity building. So this is like granting PhDs for people in other countries such that they might go back to those countries and be able to do the same things that we can do here. And so that is one of the things that I want to do. So I have my first two students arriving in my lab this year. This is Michael Orellana from Chile. He is super interested in human genomics, and this is Nick Hartman. Nick actually used to study salamanders, so I think there's a connection there. I'm very interested to go back into my like frog studying days. Um, but I also hope in the future just to bring in more Costa Rican students and more students from any other country where getting a PhD might be really difficult. So definitely reach out to me if that's something that you want to do.
Well, you heard it here. Reach out to him. So now let's take some questions from our viewers. We have a bunch of questions. So let's start with Alicia. Alicia is curious about indigenous genes from people below sea level.
Oh, okay. I need to understand that a little bit. So I know that there are parts of the world that happen to be um, sort of sunken in to the point where they're below sea level. So you have a little more oxygen there. But I never really thought about that. So that's fascinating. Like, are there specific adaptations when you have the opposite problem? Um, I don't know. Thank you for the question.
Okay. Well, thank you, Alicia. Our next question comes from Granny H. Do you miss the field or lab?
Um, so I still have friends who do field work, and sometimes they invite me to come over, and I love doing that for like 2 days. And then I remember that their lives are really hard and they work super hard, and there's mosquitoes, and you're dehydrated all the time, and you're exposed to the sun, and you don't sleep really well. So then after about a week, I'm ready to go back to my computer. In the lab, you're a primary data producer, and I'm really thankful for everyone who produces data that I didn't get to take advantage of. But I remember those were extremely long days. Like I would go in the lab before the sun was even up, and then I would leave the lab and it was night again. So then I would just miss sunlight for the entire day. Um, so I don't really miss that. I think my life is very cozy right now on a computer. Um, but it's a good question.
Well, thank you so much. Our next question comes from Pete. Can DNA tell us anything about Neanderthal vision?
Ooh, so that's a good question because I never looked into it, pun intended. But so I now wonder about, right, because we know a whole lot about the connection between like opsin genes or rhodopsin genes into vision, right? Into like the proteins that let you see in color, the proteins that also let you see in low light. And I never even thought about looking at Neanderthal opsins to see if they're identical to humans or not, because I know that they're extremely variable in like primates, for example, like New World monkeys have all this variation where they're essentially like dichromatic, but they are like, you can see where natural selection is trying to take them into trichromacy because it's really important for anyone who eats fruit. So yeah, I wonder about Neanderthals. That's a great question. I definitely will look into it now.
Our next question comes from John. And thank you, Pete. Um, you'll be looking at environment in the future to see how it may impact the spread or removal of genes from the populations.
Yeah, this is one of those questions that are like, they're very like hypothetical, right? So in the future, what does that mean for humans? Um, I don't know. I will tell you that we are definitely still evolving. None of our like modern comforts really have removed a lot of these pressures completely. We still suffer from pathogen infections all the time. Like our modern medicine is getting a lot better, but we still suffer from that. We still need to adapt to that. We, you know, the environment may be becoming more extreme now, and that means that we will have to adapt biologically as well as culturally at it. So I don't think that's going to remove genetic diversity and archaic diversity anytime soon. If anything, it might become more important. So I don't know, but that's very hypothetical. So, but thank you for the question.
All right, what is our next question? Let's see here. Okay, Penelope asks, can DNA test now find the occurrence of Denisovan and Neanderthal in each of us?
Yes, absolutely, because Neanderthal and Denisovan segments of our genomes, they are so different from the average like human genome that we can easily go look at these differences, right? The same way that I was looking at heterozygosity, like you can find portions in your genome that are very, very divergent relative to the rest of it. And so that's how we identify these segments in the first place, actually. Like before we even have access to the Denisovan and Neanderthal ancient genomes, we would just look at living people genomes, and then we say like, okay, this portion is like, this is really like these two chromosomes are so different, but only for this one little piece. So this clearly is archaic in origin. Now 23andMe, they have identified specific positions where they know what the Neanderthal variant looks like, so you can get an estimate of how much of your genome is Neanderthal from a test like that. um, to my knowledge, they don't do Denisovan um, SNPs yet, but they certainly are different enough that you can identify them. So I suspect that might become in the works for the future. But yeah, you can absolutely identify portions of your genome that are Neanderthal and Denisovan.
I can't believe how time has flown by. We have time for one more question. Um, is this mutant variant helpful to eat in eating some specific food availability, mostly in Mexico, something that is not eaten in Europe?
So that's a good question. I literally been spending the last few days meeting with a lot of saliva experts and mucin experts that can tell me a little bit more about function. And what I have found out so far, extremely preliminarily, um, no one knows. So MOG19, no one knows. It's expressed in salivary glands, expressed as in like it's your DNA makes it into at least RNA, but the protein itself is not found in your saliva, most people think. Um, so it might be that it's doing something else, like it's coding, it might be coding the cells in your mouth. And then someone proposes like, oh, do you think this is related to eating, you know, something really like challenging, right? Because a lot of these mutants, what they do is they actually coat the cells and they protect them from form a barrier to protect the cells. But it's tricky because like there's so many different environments in the American continent, and we know that Denisovan bone is found very commonly in all populations, not just in Mexicans. In Mexicans, it just happens to be one of the populations we have good samples for, so we know it's very common there. Um, and so then I have to think of like what is different about the American continent specifically about the things you eat. Like, I don't know. I know the Solanaceae, which is the family, the nightshade family, is an American family. So, like, all peppers and, potatoes and some other things um, are in this family. They have a lot of secondary metabolites that are challenging to process. Like, you have to detoxify them when you eat them. So, maybe it's related to eating um, something in the Solanaceae family, which is extremely common, throughout all these biomes. I don't know. So I'm at the like early stages of thinking about these questions, and I'm still don't have very good ideas so far. So
Okay, the last question we will ask is, do you have any advice for those who are just starting out or hoping to pursue a career in science?
yeah, I would say like if this is your passion, chase it. Don't give up, don't settle. Um, reach out to people. Like that's what we're here for. We're here to help. Like we want to see people like ourselves and just help them get to where we are. So, reach out to people, ask for help, ask questions, and just don't be discouraged. It's a difficult road. Um, it can be difficult to navigate, especially if you're coming from outside the US or Europe. But everyone around the world has a lot to contribute. So definitely, just ask for help.
That is absolutely excellent advice. And thank you so much for being here with us today.
Thank you so much for having me. Thank you for everyone who's watching.
Well, next time on Lunch Break Science, we'll be featuring Leaky Foundation grantee and recipient of the 2023 American Association of Biological Anthropologists and Leaky Foundation Communication and Outreach Award, Dr. Kara Akabach. It's going to be a wonderful episode, and I hope you will all join us for it. Well, thank you all for taking a break from your day and feeding your brain with the Leaky Foundation. Until next time, stay hungry for knowledge. Lunch Break Science is brought to you by the Leakey Foundation and made possible by the generous support of the Ann and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund, as well as viewers like you. Show your support of Lunch Break Science by subscribing to our channel, clicking on notifications, and giving us a thumbs up, or making a donation to help us create new content. Still craving science and can't wait for the next episode? You can feast on the Leaky Foundation's content library with past episodes, lectures, our podcast origin stories, and more. Thank you all for tuning in, and see you next time.